In this lesson we will present to you chemistry, manufacturing, and controls dossier overview. The lesson will consist of the following parts: lesson objectives, definition of terms and abbreviations, a short introduction, CMC overview, CMC sections, chemistry, manufacturing, and controls. Lesson objectives. After this lesson you will be able to describe the main sections of Module 3 of the Common Technical Document. Define the key terms relating to chemistry, manufacturing and controls. Be able to discuss the organization of the chemistry section of Module 3 dossier. Be able to discuss the organization of the manufacturing and control sections of Module 3 dossier. Learn why sections of Module 3 vary with drug product. Be able to describe what can delay compilation of Module 3. Be able to discuss the regional requirements of Module 3.
Definitions of terms and abbreviations. In this section we will define a few technical terms that are found later in this slide presentation.
These are microorganisms or biological agents that are not deliberately introduced into the manufacturing process of a biological product. They include bacteria, fungi, mycoplasmas, rickettsia, protozoa, parasites, TSE, BSE, and even viruses. API and dosage form. Active pharmaceutical ingredient (API) is a substance intended to furnish pharmacologic activity or other direct effect in the diagnosis, cure, mitigation, treatment, or prevention of disease or to affect the structure or any function of the body. Dosage form contains an active drug ingredient or placebo. Batch. A batch is a quantity of drug in dosage form. A raw material. Or a packaging material, homogeneous within specified limits, produced according to a single production order and is attested by the signatories to the order. In the case of continuous manufacture, a batch corresponds to a defined fraction of the production that is characterized by its intended homogeneity. It may sometimes be necessary to divide a batch into a number of sub-batches, which are later brought together to form a final homogeneous batch. Batch records. Batch records are detailed description of pharmaceutical manufacturing processes. Biologics. They are a wide range of products such as vaccines, blood and blood components, allergenics, somatic cells, gene therapy, tissues, and recombinant therapeutic proteins. They may be composed of sugars, proteins, or nucleic acids or complex combinations of these substances, or may be living entities such as cells and tissues. They may be isolated from a variety of natural sources including human, animal, or microorganism. They may also be produced by biotechnological methods and other technologies and may be used to treat a variety of medical conditions for which no other treatments are available. Biosimilars. These are biologic health products which are almost identical to an original product that is manufactured by a different company. Compendial. Compendial means related to a compendium which serves as a standard. A compendium includes United States Pharmacopoeia, British Pharmacopoeia, European Pharmacopoeia, and many others. It is used to refer to any of the pharmacopoeias. Generics drugs. These are drugs that are identical within an acceptable bioequivalence range to their brand name counterparts with respect to quality, pharmacokinetic and pharmacodynamic properties. The standards and requirements for approval of generics may vary from country to country. Overages. Overages refer to approved addition of an active pharmaceutical ingredient more than 100% above label claim during the dispensing of ingredients to compensate for loss during the manufacturing process of the drug product and to meet the label claim. Pharmaceuticals. These are compounds manufactured for use as medicinal drugs. Phase AB and Phase 3 IND. Phase AB clinical trials are well controlled and are designed to evaluate efficacy and safety of a drug candidate in patients with the disease or condition to be treated, diagnosed, or prevented. These clinical trials usually represent the most rigorous demonstration of a medicine's efficacy. They are sometimes referred to as pivotal trials. Phase 3 IND are clinical trials conducted after efficacy of the medicine is demonstrated, but prior to regulatory submission of a new drug application (NDA) or other dossier. These clinical trials are conducted in patient populations for which the medicine is eventually intended. BSE and TSE. TSE is transmissible spongiform encephalopathy. BSE is bovine spongiform encephalopathy. Validation. Validation is the documented act of demonstrating that any procedure, process, and activity will consistently lead to the expected results. Includes the qualification of systems and equipment. Introduction.
In this section I will introduce Module 3. The terms Module 3 and CMC will be used interchangeably. In this section we will learn where CMC fits in the common technical document. You will also learn the main sections of the CMC. Chemistry, manufacturing, and controls in a dossier is also referred to as Module 3 and is part of the CTD. It consists of Quality Overall Summary, COS, in Module 2 and Module 3 Quality. COS is a summary of Module 3, but it's organized in a modular format. There are different section headings and numbering. See next slide. Module 3 for a dossier to obtain market authorization requires the following key sections: drug substance, drug product, appendices, regional information, literature references. Main sections of Module 3. Sections of CMC. Nomenclature. Drug substance. Drug product. Appendix. Facilities information, adventitious agents, excipients. Regional. CMC may be divided into 3 main sections: chemistry, manufacturing, controls. CMC content. CMC content is variable and is based on product type, for example pharmaceuticals, biologics, vaccines, biosimilars and generics. Level of detail depend on product type. Regulatory agencies require less content for pharmaceutical products than vaccines and other biologics, perhaps because they are not well characterized. Who needs CMC? The following FDA applications require CMC. Investigational New Drug Application, IND. Phase 2B and Phase 3 IND. New Drug Application, an abbreviated New Drug Application. The following applications to Health Canada require CMC. Clinical Trial Application, CTA. New Drug Submission. Abbreviated New Drug Submission. Level of detail in CMC depends also on the country. Each country has its own CMC requirements. Check the regulations and guidance documents for your country. In USA, the level of detail depends on the stage of drug development, such as IND. This requires the least content. Phase 2B and Phase 3 IND. Marketing Authorization Application requires the most content. Post-marketing requirements varies and it depends on the CMC section or subsection. CMC Overview. In this section I will make a brief overview of the CMC. Sections of the CMC in drug product and drug substance. There are 2 major sections in the CMC dossier, namely drug substance and the drug product. The following sections of the drug substance are required: general information, manufacturing, drug substance development, characterization/impurities, control of drug substance, specifications, description of assay methods, assay validation, batch analysis, justification of specifications. The following sections of the drug product are required: description of the drug product, manufacturing, control of excipients, their specifications, description of assay methods, assay validation, batch analysis and justification of specifications. Sections of the CMC in drug product and drug substance. This slide is a continuation of the previous slide. The drug section relating to the drug substance will also include reference standards/materials and container closure system. In the stability section you should include stability data summary, stability commitments, Stability data and control of drug substance. The section on the drug product should include under control of the drug product the following: specifications, description of assay methods, assay validation, batch analysis and justification of specifications. The section should also include reference standards and container closure followed by stability data, which includes stability summary, stability commitments, and stability data. This slide shows that the drug substance and drug product could be either a solid or a liquid. Facilities information. This section relates to biotechnology products. Diagrams should be provided to illustrate raw materials, personnel, waste and intermediates flow in or out of the manufacturing areas. Provide information on other products that are manufactured in the area, including lists of products and type, and manufacturing schedules. Description of product contact equipment, including cleaning information for single and multi-use equipment such as tanks, lines, fermenters and bottles. Information on design features to include area classification such as Class A, and features to prevent cross-contamination. Here you provide the information to show how the following agents have been excluded: TSE and BSE, bacteria, mycoplasma, fungi, viral. Provide viral evaluation studies relating to materials of animal origin such as milk, hair, hooves, bones, bovine serum albumin. Materials of human origin such as blood and organs. Viral clearance studies data may include data that demonstrate the validity of the small-scale process when compared to the commercial-scale process. Applicants may be required to discuss the adequacy of viral inactivation or removal procedures for manufacturing equipment and materials, and manufacturing steps that are capable of removing or inactivating viruses. Regional section of the CMC. Some regions have their own specific requirements. USA requires the following: environmental exemptions for IND, executed batch records for BLA. Regional section of the CMC. EU requires the following: viral safety dossier, C/BSE declaration, GMP certificates for materials of animal origin. Canada requires executed batch records.
CMC sections. Quality sections. The chemistry section describes the composition of the drug product and drug substance. Manufacturing section describes how the manufacturers make the drug product and drug substance. The controls section describes how the manufacturer ensures that the drug product and components meet preset specifications of the product. Chemistry. This section is about the chemistry of the drug substance and the composition of the drug product. This section varies with the chemistry. This chemistry section consists of the following: general information about the drug substance, characterization of the drug substance, composition of the drug product. What is included in general information? We have nomenclature includes compendial names, chemical names, laboratory names, other non-proprietary names. Structure. You should include the following: for new chemical entities, stereochemistry, molecular formula, and molecular mass. For biotechnology-based products, amino acid sequence, protein structure, post-translational modification, nucleic acid sequence. General properties of drug substance. The following needs to be included. For new chemical entities, provide physicochemical and other relevant properties. For biotechnology-based products, provide biological activity. S1.1 Nomenclature. This section is variable and includes compendial names, chemical names, laboratory names and other non-proprietary names. S1.2 Structure. This section is variable as it depends on the particular drug. What to include under general properties. Elucidation of structure and other characteristics. New chemical entities. Here you confirm the structure and stereochemistry and describe potential polymorphs. Biotechnology-based products, provide primary if possible, secondary, and higher-order structure, post-translational forms, purity, biological activity and immunochemical properties. Impurities. This includes anything that was not added intentionally. Impurities. These are found in raw materials or finished products, however they are not added. Information on impurities will depend on the drug substance. Description and composition of drug product. Provide a description of the drug product and its composition. The information provided should include, for example, description of the dosage form. Composition, that is, list of all components of the dosage form, and their amount on a per-unit basis, including overages, if any, the function of the components, and a reference to their quality standards, for example, compendial monographs or manufacturer's specifications. Description of accompanying reconstitution diluents if relevant. And type of container and closure used for the dosage form and accompanying reconstitution diluent, if relevant. Manufacturing. In this section we will consider the sections of the CMC that describe the manufacturing of the drug substance and drug product. What to include in the manufacturing section of CMC. Manufacturing consists of materials, manufacturer, manufacturing process, manufacturing process validation, manufacturing process controls, development of drug substance and drug product, container closure for drug product and drug substance. Manufacturing process. Excipients. The drug product consists of drug substance and excipient/excipients. The type of excipients in a drug product vary with the formulation. Materials. These are raw materials bought from a vendor or made in-house. For example, biologics may be made in-house, while the drug substance such as acetaminophen may be purchased from a vendor. Obtained from a vendor. Made in-house. Materials. Raw materials. Control of materials. Here you list the materials used in the manufacture of the drug substance and where each is used. Raw materials. Materials bought from a vendor. Starting materials. Materials made in-house. Cell banks. Master seeds. Solvents. These are substances that dissolves or solute. Reagents are substances or mixture that are generally used in chemical analysis or other reactions. Catalysts. Substances that increase the rate of a chemical reaction without itself undergoing any permanent chemical change. Excipients. Control of excipients. Excipients are natural or synthetic substances included in a formulation. They may bulk up formulations that contain very small amounts of the drug substance. They may enhance drug absorption or slow drug release. Assist in the manufacturing process by enabling powders to flow without sticking on the surfaces of containers and other surfaces. Improve stability over the expected shelf life. You provide a brief summary on the quality of excipients. Manufacturer. Here you provide address for the site responsible for manufacturing, testing, packaging, and releasing the drug product. Manufacturing process validation. Provide description, records, and results of the validation and/or evaluation studies for critical steps or critical assays used in the manufacturing process, for example, validation of the sterilization process or aseptic processing or filling. Viral safety evaluation should be provided if necessary by assessing filter qualifications, critical equipment and component qualification, process simulations, media challenges, shipping qualification. Manufacturing process controls definitions. Critical steps. Provide tests and acceptance criteria with justification including experimental data performed at critical steps identified in the manufacturing process to ensure that the process is controlled. Intermediates. Provide information on the quality and control of intermediates isolated during the process should be provided. Manufacturing development. Manufacturing process development. In section you describe the changes, if any, that you made to drug substance during scale-up. P2 Manufacturing Process Development. Here you describe the changes, if any, that you made to drug product during scale-up. Development. Manufacturing Process Development. Changes made to drug substance during scale-up. Pharmaceutical Development. Components of the drug product. Drug substance. Discuss the evidence for compatibility of the drug substance with the excipients. Excipients. The choice of excipients, their concentration and their characteristics can influence the drug product performance. They should be discussed relative to their respective functions. Development continued. Drug product. Provide a brief summary describing the development of the drug product. Include formulation development, overages, physiochemical and biological properties, manufacturing process development, container closure system, microbial attributes, compatibility of the different components. Container closure. Drug substance. Stainless steel tanks are used for liquids. Secondary packaging is what the product or primary packaging is stored or shipped in and includes trays, cartons, boxes. Container closure. Whatever touches the drug substance is included as container closure. It may be a liquid, powder or crystals. Typical storage containers: stainless steel can, bottle, bags, barrels. Storage conditions include room temperature, refrigerator, freeze. Container closure. Drug product primary. Primary packaging material touches the product and includes ampoules, blister packs, bottles, caps, syringes, plungers, vials, caps, seals. Container closure. Specifications. Description, for example, dimension, characteristics, compendial compliance, United States Pharmacopoeia, European Pharmacopoeia, Japanese Pharmacopoeia. Identification, for example, give the name and manufacturer is required. Provide critical dimensions of the container closure with drawings. Controls. In this section you will learn the type of controls that should be presented to regulatory agencies as evidence that your company has procedures in place to assure future production of a product with reliable quality. Significance of controls in CMC. This section is very important, and it includes the following: control procedures to assure product consistency and quality, identification of quality attributes that characterize the product, appropriateness of the test methods used to monitor product quality, evidence for how long the product will maintain its quality after it is made. That is the shelf life. Controls. Outline. Specifications. Provide actual specification of the drug substance and the excipients. Provide the justification of specifications for the excipients and the drug product. Assays. Provide a description of the analytical procedures used for testing the excipients and the drug product should be provided, where appropriate. Validation information. Provide analytical validation information, including experimental data, for the analytical procedures used for testing the excipients and the drug product. Reference standards. Batch analysis. Characterization of impurities. Stability testing. Specifications. Here you provide a list of drug substances or drug product requirements. For the specifications of a drug substance and product include a list of tests, references to analytical procedures, acceptance criteria with numerical limits, ranges or other criteria. Provide the specifications for the excipients and the drug product and the justification of the same specifications. Assays. Assays, how materials are tested and how we know that the test is accurate. Description. Describe how the actual testing is performed. Validations. Provide information on what parameters were checked to make sure the information is accurate. repeatable, and reliable.
Validation and computible methods. Validation proves the assay is reliable with people, labs, and different lots/batches. If the assay is performed as described in a pharmacopoeia, that assay is considered validated. Confirm there are no components that would impact assay performance. Validated with another product to ensure assay performs as expected. Validation proves the assay is reliable with people, labs, and different lots/batches. Reference standards. These are materials used in an assay. They must have known concentration. Results of known standards should be used to calibrate equipment and predict values for unknown samples. Stability. Stability information includes stability summary and conclusions, post-approval stability protocol and stability commitment, stability data. Stability summary and conclusions. Stability summary and conclusions. Types of studies conducted. Protocols used. Provide results of studies summarized with respect to storage conditions and retest or shelf life. You may use H studies including forced degradation and stress conditions. Stability data. Results of the stability studies should include a list of tests that have been done, acceptance criteria, initial batch release results, testing time points and data if available. Batches for stability testing will typically include pivotal batches used in clinical trials, clinical lots, important safety assessment lots. Formal stability studies, these are lots used to support shelf life. Used for marketing critical lots to support marketing. Accelerated stability testing includes stability data for all batches that are included in the batch analysis. Post-approval stability protocol and stability commitment. Post-approval stability protocol and commitment includes a protocol and a commitment to continue the ongoing stability studies to support the shelf life of a product. Stability protocol and commitment. This section provides the post-approval stability protocol, stability commitment, and ongoing stability studies for drug product manufactured at the commercial manufacturing site. The data used to support the proposed expiry at the long-term storage conditions should be evaluated against the proposed commercial specifications. Factors that delay the completion of CMC. Team members. The number of people dedicated to the project will determine how long it will take to complete the project. There should be adequate number of people working on the submission to avoid delays. Experience of the team members is also important as lack of experience and knowledge can cause unnecessary delay. Factors that delay the completion of CMC. Lack of relevant information from contract manufacturing organization, contract research organizations, or in-house can delay the completion of CMC section. The following are the common sources of information: manufacturing site and packaging site among others. Common factors that delay completion of CMC. Lack or inadequate data such as stability studies, process validation, validation studies, adventitious agent testing, characterization including impurities, and many others.
Thank you for listening. See you in the next lesson.
This lesson was brought to you by Royal Canadian College of Distance Education.